So we're going to start up our next section looking at some of the mechanisms that some plants have to ensure as much as possible that they outcross as opposed to self-pollinate. So for some plants, plants that are monoecious, and monoecious is kind of like being hermaphroditic if you're an animal. They've got both male and female reproductive organs. For a monoecious plant that doesn't want to self-fertilize, they need to have some way of separating in space male and female flowers. And so the most common thing would be to have a plant that's monoecious, both male and female, having the female flowers located toward the top of the plant and the male flowers located further down toward the bottom of the plant. And that's going to, as much as possible, prevent pollen grains from one plant arriving on the female flowers of the same plant. For dioecious plants, those that are either male or female, not hermaphroditic in other words, then that's not an issue. They actually have separate male and female plants, so they obviously cannot self-fertilize. Another possibility is to have what's called self-incompatibility, where even if pollen from a plant lands on female flowers from the same plant, there is no fertilization. So there could be chemical or genetic ways of preventing a zygote or an embryo from being formed if the pollen is too similar of a match to the egg in the embryo sac. There can also be what's called a dichogamous situation, which is where you have temporal separation. So in other words, you might have male flowers that mature in May and female flowers don't mature until, say, June. So that's going to help also prevent fertilization on the same plant. So, again, looking at an individual plant, the male flowers on that one plant might mature in 1 month, and the female flowers might mature a month later or 2 weeks later. So there's no real way that the pollen from that plant can fertilize flowers on that same plant. Now, within the population, then, obviously there has to be some overlap, but each individual plant would be somewhat different in terms of when their male or female flowers might mature. Now, double fertilization is the hallmark of the angiosperms as a group of plants. It is something that only the flowering plants do. It's a very, very advanced type of reproduction, and it's one that oftentimes the first time you see it is a little bit overwhelming. Not to scare you, but there's kind of a lot of detail to how this process actually works. So we're going to go ahead and go through it. There's a couple of things that happen here with double fertilization. There's going to be the actual fertilization event where a sperm cell reaches an egg cell and creates a zygote. And there's going to be a secondary event that's not technically a fertilization because there is no zygote that's actually produced. It's more of a fusion of a sperm cell with those two polar nuclei cell that we saw a couple slides ago, or in the previous section a couple slides ago when we saw the embryo sac. There were two polar nuclei plus one nucleus from a sperm cell is going to produce a triploid. So now haploid is one set of chromosomes, diploid is two sets of chromosomes, triploid is now 3 sets of chromosomes that are produced in this one structure. And what that does, the endosperm actually becomes the nutritional reserve for the embryo as it's developing. It's like an energy reserve for the embryo as it develops. So sperm cell plus 2 polar nuclei equals triploid endosperm. All right, so now we have a new kind of a structure that we haven't seen before, and really no other organisms except for the angiosperms do this. With fertilization comes the production of a zygote. Fertilization of the egg produces a zygote, and then fusion of sperm cell with polar nuclei is going to create the triploid endosperm. So that's where the double part of fertilization comes in. Even though it's technically not double fertilization, there's really only one fertilization because there's only one egg cell involved. There are 2 sperm cells involved, so some people would call this double fertilization. All right, so here's kind of the progression of events. We have our pollen grain over here, which is, by the way, not to scale. Pollen is nowhere near this big compared to the female parts of the flower. It's tiny. But there's a tube cell that's going to produce our pollen tube eventually over here. And we have a generative cell, which is going to produce the 2 sperm cells that will eventually fuse with the polar nuclei and the egg cell. So the pollen grain is going to land on the stigma. There's going to be the growth of the pollen tube down through the style. And you can see here in the pollen tube are these 2 sperm cells that were produced by the generative cell. And so now over time the pollen tube grows further and further down through the stigma, around the ovary, up through this opening here called the micropyle. And at that point the 2 sperm cells will be released into the embryo sac. One of the sperm cells fuses with the egg cell, the other sperm cell fuses with the 2 polar nuclei, and so now we have our diploid zygote and our triploid endosperm. Okay, so that's the double fertilization. It seems to be fairly complicated, but once you kind of walk through it a couple of times, it's actually fairly straightforward. It's more complicated than, say, what an animal does, for example, but it is still fairly logical in the terms of progression of events that have to take place. Now, looking at asexual reproduction, this is now where plants are going to be producing genetically identical individuals. And so this is going to now ensure that there's not going to be any meiosis, right? Because to have meiosis means you're producing gametes. When you're making gametes, you're reproducing sexually. This is asexual only. So it's going to be strictly mitosis. This is going to be most common in very, very severe environments, because if a parent plant is able to survive in a severe environment, then clearly they're doing something right from a genetic standpoint. And mixing their genes together with genes from somebody else could be kind of risky. Your chances are going to produce a bunch of offspring that maybe aren't that well adapted to the environment. So you're better off to clone yourself, make all the offspring like you, because after all, you're surviving. And if your offspring are just like you, then they should be pretty good as well. Any variations that occur as a result of meiosis or sexual reproduction, chances are, if the environment's harsh, those new traits are not going to be adaptive, and they're only going to be harmful. There is also a situation that some plants do called apomixis, which is where you actually get asexual development of an embryo inside of an ovule. So this is a way to produce a zygote and eventually an embryo without actually having sperm cells present, which is kind of a unique thing, and actually something that's very useful, because you still get seed dispersal. These plants still are able to produce seeds, and they're still able to produce fruits, which we haven't talked about yet. And as such, they get the full benefit of having the seeds produced inside of fruits, and having those fruits dispersed perhaps by animals or by wind or by water. Again, not all plants can do that, but it is an option for some plants. There's actually a lot of interest, too, in doing this in agriculture, because we could actually have fruit production and seed production without actually going through the pollination process. It would make things a lot easier. You wouldn't actually have to go around and pollinate plants with insects, or even doing it by hand for some species. There is also vegetative reproduction, and so this would be where you have plants that break off of a parent plant and start to grow. And so we actually saw a picture of this back, I think, in our previous chapter, the calancho plant that has these very, very little tiny, what are called plantlets, little tiny miniature versions of the adult, that if these were to break off, if they hit the soil in the right spot, they can actually start to grow, and they will be genetically identical clones to that parent plant. We've seen runners and stolons before, whether they're above ground or below ground. These are sort of like horizontal spreading reproduction, and things like strawberries and those kinds of things. We've seen rhizomes before. We've seen suckers, where a plant will send up sprouts through the soil around itself that are very small versions of itself, still genetically the same plant, but it's a way for them to kind of colonize out away from where the one primary plant actually is. And then these are plantlets. These are actually called adventitious plantlets, growing on the leaves of this calancho plant. And again, when those break off, they will actually start to grow anew. You can actually clone entire plants using a nutrient medium, and this is what actually is done in laboratory situations where you want to grow a very, very conservative line of plants. You want to grow the same genotype over and over and over quickly. You can actually grow cells. You can start with an individual cell or even tissues on nutrient medium, and then very quickly kind of grow up samples that way. So what actually happens here is you take that cell, you take away the cell wall, and what you're left with when a plant cell has a cell wall removed, it's referred to as a protoplast. So think all the way back to chapter 3 where we talked about cells. This would be a plant cell minus a cell wall. You would have the cell membrane, the plasma membrane, the cytoplasm, the nucleus, all the other stuff inside the cell. You're just missing the cell wall. And so once you do that, the plant's tendency then is to be able to differentiate itself into different types of organs, or even the entire organism. Basically what you're doing here is you're creating a plant stem cell. Not a plant stem like a trunk, but a plant stem cell, like a cell that could become any other part of the plant. So you can take cells from a leaf, or cells from a stem, or cells from a root, and remove that cell wall and sort of encourage them to become an entire plant into themselves as a result of this plant culture technique. Eventually what occurs is the cells in the culture just divide and divide and divide. They form what's called a callus, which is a mass of plant tissue. And then at that point, you can either use the callus as needed, or continue to drive the plant down the pathway of becoming a fully functional individual independent plant by itself. Now, as far as lifespans of plants, this is kind of interesting because, again, plants work very, very differently from how animals do things. Plants, for example, never die of old age. That's not a thing if you're a plant. They can live for a really, really, really long time, thousands of years. There are plants alive now that have been alive for literally 4,000 to 5,000 years. A lot of plants only live for a single year. Some plants only live for 2 years. Most of the woody plants, those that can do secondary growth, live for many, many years, decades, centuries, even millennia in some cases. So think about a plant that's been alive for 5,000 years. The kinds of things that have happened on our planet in the last 5,000 years, right? It's amazing to think about what's happened during the lifespan of those plants. Generally, though, the way we classify plants in terms of life cycle is based on how long they live. So annual plants, if you know anything about gardening or landscaping, annual plants complete their entire life cycle in a single growing season. So the seeds germinate, they grow, they mature, they flower, they reproduce, they die, all in the span of a single growing season. Maybe even less than a year. If you live in an environment like we do in Northeast Ohio, it's not even going to be a full calendar year. It's going to be a few months before it's gone. If you have a biennial plant, these plants are going to take 2 years to complete their life cycle. So what happens for these? They're going to germinate in the first year, seeds are going to sprout, they're going to start to grow, and then during the first winter of their life, they're going to go dormant. They're going to kind of shut down, they're going to more or less go into what amounts to hibernation. The following year, they're going to sort of reactivate themselves, they're going to finish their growth, they're going to reproduce and flower, and then they're going to die toward the end of the second growing season. For perennial plants, these are plants that keep coming back year after year. And for these plants, there really is not a finite lifespan for them. Typically with most plants, longer lived ones, perennial plants, it's usually a matter of something killing them rather than dying just because they get old, right? So it's usually a matter of an herbivore coming through, a disturbance like a fire coming through or a drought coming through, some other sort of outside influence that actually takes the plant out, as opposed to the plant sort of breaking down physiologically and just failing to function like animals do as they age. Now, once we have everything we've done so far has been either sexual reproduction up to the point of fertilization or asexual reproduction. So what we're going to pick up with now is we're going to take a look at what happens once the egg is fertilized. So now we have a zygote, we have a fertilized egg. We also have the endosperm, right? So the surrounding part here, these two polar nuclei, they're going to fuse with the other sperm cell, are going to become the endosperm. So we're going to have triploid endosperm, diploid zygote. What happens next? All right, so at this point we have a zygote, and as soon as that zygote divides, we're going to have an embryo. A 2-cell initially, but then eventually more cell embryo. So we've kind of seen the double fertilization thing. This is a bit of a review. I actually took this from a different lecture. I put it together with a previous part that I made here. We have the pollen tube dropping off the 2 sperm cells, fertilizing the egg, fusing with the 2 polar nuclei to form the endosperm. So we have the nutritional source, and we also have the embryo. So now what we're going to look at now is, okay, what happens with the embryo? What does the embryo do next in order to get to the point where it can start to produce a seed and eventually start to produce a fully independent plant? So initially, early on, what's going to happen here is the zygote is going to start to divide, but it's going to divide asymmetrically. Right? There's going to be 2 ends to this zygote, what eventually becomes an embryo, and they're going to have 2 very, very different fates or destinies or places where they're going to go eventually. Right? So there's going to be a relatively small area of cells, more or less a spherical shape, which is the embryo itself. It's the smaller part of the structure, is the embryo. The other part is a larger group of cells that divide to form what's called a suspensor. Now, the suspensor is what eventually is going to take on the appearance of something like a root. Now, it's not actually the root itself, but the suspensor is the part that connects the embryo to the endosperm. Right? It's connecting the embryo to the endosperm. Think about this as being kind of like an umbilical cord in a mammal. It connects the embryo to the parent plant. In this case, the endosperm part. So this is the conduit through which nutrients can go from the endosperm into the growing embryo. So that's our nutrient part of things. At this point in time, again, very, very early, these embryos are only a few dozen or maybe a couple of hundred cells in size. But even this early on, there's going to be a very good indication of where the root shoot axis is going to be. So in other words, the long axis of the plant, we'll be able to see where that's going to come into play as well at that point in time. The cells closest to the suspensor become the root. Again, these are not the suspensor cells themselves, but they're close to it. The part of the embryo closest to the suspensor, those cells are going to become roots. The other end is going to become the shoot. So we actually have now a bit of directionality to our very, very young plants, and it's starting to show the earliest signs of differentiation into some of its later structures. And so again, just reviewing what we had here, we had the sperm cells dropping off, polar nuclei up here, fertilization of egg, fusion with polar nuclei, triploid endosperm, diploid zygote down there toward the bottom. So here's our cell division, right? We've got our very first cell division down here. We're going to have this green area up here is the endosperm. this pair of cells right here is the result of what the zygote was. So if we go back, right, here's our zygote, single cell, but it's diploid. This cell is going to divide, but it's going to divide asymmetrically. So what it's going to produce is kind of like this little snowman shape here, where this smaller cell eventually is going to give rise to all the cells that become part of the embryo, and this bigger cell is going to give rise to all the cells that become part of the suspensor. Okay, so we've got endosperm, embryo, suspensor. Endosperm, embryo, and then suspensor down here. And so separately, but still kind of in kind of the same realm of time, the suspensor cells and the embryonic cells are going to start to divide. So as time goes on, we now have more of a globular or spherical shaped embryo. These cells here are going to become the roots eventually. And we also, of course, have our suspensor, which is dividing maybe not quite as fast, but these cells are still dividing as well. From there, we then go into the next step, further cell division. Now we actually have cotyledons, and cotyledons are the indication of the very, very first leaves that plant is ever going to have. If you think back to some of our previous sections, we talked about monocot versus dicot. Right? Cot is referring to cotyledon. This plant would be a dicot, or dicotyledonous plant, because it has two cotyledons. If a plant just has one of these, then it would be a monocot. Right? So if you look at the number of seed leaves a plant has, that's also a good indication of whether they're monocot or dicot. Eventually, these cotyledons get bigger. We start to see more differentiation here into a stem and into roots. We start seeing primary tissue types. So we've got protoderm. Remember, protoderm eventually gives rise to dermal tissue. We have ground meristem up here in the middle, which gives rise to ground tissue. And then here in the middle of this, the orange area, this is the procambium, which eventually gives rise to the vascular tissue of the plant, the water conducting and sugar conducting parts of the plant. As this gets even bigger, now we can start to see the shoot apical meristem. The root apical meristem is down here. We've seen those. Those are the places that are going to provide the first growth in both directions, up and down, for the shoot and roots respectively. We've got both apical meristems in place. We also have a section of stem called a hypocotyl. Hypocotyl is a part of the stem below the cotyledon, so this is the hypocotyl. And again, we still have our cotyledons. If you look at what's happened now to the endosperm, at this point in time, it has been largely depleted. A lot of the energy present in the endosperm has been used up by the embryo in the process of growing, because all of these cell divisions, to go from 2 to however many cells or each one of these other stages, all those cell divisions have to be fueled by energy. And so the energy in this case is coming from the endosperm, and there's, in this case, in this picture, not that much endosperm left. All right, so I'm going to cut off here, and we'll start up the next section momentarily.